And then for our last talk of the session, as well as the symposium, we have with us Hans Hoffman from UT Austin, where he is a professor of integrative biology. Hans has been widely known for a number of years for studying cichlids, putting cichlids, sort of establishing them as a new model organism. He's brought a number of genetic, genomic, and neurobiological tools to these fish. But he assured me that he will not only talk about cichlids, he will finish on more recent work that ties into the question of this conference. You are not ready,
Thank you.
Are you?
It's doing something. Okay, thank you. Thanks, Felix. And it's just starting up. It's okay, it just takes a moment. Yeah. I don't know why, but I might as well get started while this is doing its thing. It says, please wait. I'm the only thing that's standing between you and your lunch. And I don't want to delay that too much. So what I'm going to do is I will talk to you about two different things. But first, I want to thank the organizers for putting on this amazing symposium and for giving me an opportunity to talk to you. And I'm a neuroscientist. Back in the olden days, I was trained as a neurophysiologist. I went to the dark side and went from invertebrates to vertebrates, from electrophysiology to molecular biology, and then to omics. Some people never forgave me for doing that, but oh, this is bad.
You got it. You're logging in now.
Yeah. Well, I'm not sure why.
I think it's good.
Yeah, but why would it even do that? Must be in a union. And pardon me? No, it's doing it's just really slow. Yeah. Okay. Don't get excited too early. So, okay. So, as I said, I am a neuroscientist pretending to be an evolutionary biologist. And what I want to do is tell you a little bit about what two different projects. Now we're on, I think. Mostly what we're interested in our lab is to understand the molecular and neural basis of decision-making, all kinds of different decision-making in different contexts. And we study this in different species. We've worked on more than 120 species, of course, with a lot of collaborators. In the laboratory, we mostly work on cichlid fish, either from East Africa or from Central America. But what I want to do now is give you a basic idea of how I think about a particular aspect of biology that we study a lot in my lab, which is the transcriptome, can actually tell us a lot about not just things that happen in real time, not just things that happen maybe over the course of days or of the course of seasons, but also over longer time scales. And so the basic idea is that we can partition the transcriptome and its variants into flexible and constitutive properties. And so I want to walk through this a little bit for a moment because I think it's going to be important later. So the idea is that we have different time scales that I call here real time, ontogenetic time, ecological time, and phylogenetic time. In that a neuroscientist might call this modulation, a physiologist might call this plasticity, an evolutionary biologist or an ecologist might call this microevolution, and we might call this a macroevolution. And examples would be situations, and we know, and I've done this for many years, that the transcriptome is incredibly susceptible to changes in real time, to responding to stimuli of different kinds. It changes over the course of the day. It changes with development. Sex attributes develop over the course of maturation. There are seasonal changes. There are life history changes, and so on. And then eventually we get into life history, we get into ecology and social system, which also can be reflected in the transcriptome, although this is not maybe what people study as much. And we also have these more long-term signatures that we can identify that I might call constitutive aspects of the transcriptome, such as sex, as far as chromosomal sex is concerned, tissue identity, or species, and you probably can add other things to it. And so this kind of framework, which I find very useful to think about, I want to put into the context of comparison across species, and to eventually get inferences that are just as good as doing experiments. That's the basic idea. And this is not a new idea. It goes back all the way to Aristotle, which we can consider as the founding individual of modern biology, although he lived a long time ago. And I want to read this quote to you because I think it's really useful. He said, ought we, for instance, to begin by discussing each separate species, man, lion, ox, and the like, they had different model systems at the time, taking each kind in hand independently of the rest, which is what biomedical research largely does, or ought we rather to deal first with the attributes which they have in common in virtue of some common element of their nature and proceed from this as a basis for the consideration of them separately? And I think that is a very important insight. He had no idea about evolution. He got other things wrong, but I think this is quite fundamental. A modern incarnation of this sentiment comes from John Maynard Smith, one of the preeminent evolutionary biologists of the last century, who said we must learn to treat comparative data with the same respect as we would treat experimental results. So with this idea, let's look at this constitutive aspect for a little bit. And so what we've done recently is that we collected data from 25 species across vertebrates on the medial amygdala in the hippocampus. This is bulk RNA-seq data that we either generate ourselves or we get from public databases. We developed a universal custom pipeline for mapping and quantifying reads, and we identify one-to-one orthologs. And this is work by a grad student in the lab, Will Hahn. And so we focus on these particular regions. There are two reasons for that. One is that the medial amygdala is important for social valence and salience and effective decision-making, and as a social neuroscientist, I'm interested in that. And the hippocampus, because it's involved in space use, learning, and memory. The other reason is that this is the data that we could get a hold of, right? There is a lot more to be done here. This is just a start. When we reconstruct the evolution of these transcriptomes, we largely, and not surprisingly, recover the phylogeny of these species. It's not 100%, it's just like a gene tree. It may not necessarily reflect species phylogeny completely, but it's pretty close. Same in the hippocampus. Now, I want to do a little step here, and I, instead of showing it this way, I'm going to show you the data in the context of a principal components analysis. And so what you see here, I'm plotting here PC2 against PC7 for a reason. And the first two principal components largely explain phylogeny, which is not surprising. That adds up to about 37% of the variance. But if I change the colors here a little bit, we actually get the tissue signal between the hippocampus and the medial amygdala. And I can add the fish to that as well. We find this difference as well. It's just when I show them all together, then the other species all get bunched into one corner, and you don't see the difference. Now, what we'd like to know is to which these signatures are actually similar across even distantly related species. And so we compare the medial amygdala with the hippocampus in each species to identify the genes that are differentially expressed. That's not ideal, but that's what we can do at this point. And we rank them according to the expression difference in, let's say, a mouse and in a fish. We rank them, we bin them, and then we use what is called a rank-rank hypergeometric overlap approach to identify concordant or discordant expression. So in this particular example, the genes that would be lowly expressed in, say, the medial amygdala versus the hippocampus show much more similarity and overlap or concordance than we would expect by chance between, let's say, here a mouse and a fish. And so we can do this systematically. Here, I just told you this, for, say, a cichlid and a mole rat, for example. We can then, on all these pairwise comparisons, extract the genes that are enriched in these, significantly enriched in these bins, do a Pearson correlation to get a singular metric so we can compare across the entire analysis. And then we find, for example, that cichlid and stickleback are quite similar in this regard, which may not be surprising. We can add the sparrow here, and then you might say, oh my God, they're all the same. But it turns out it's not. There is actually some variability here, but it doesn't necessarily correlate with evolutionary distance. So it's not like that the more evolutionarily close species are more similar in the expression variation between hippocampus and amygdala, but there seems to be something else going on. And so, and I'm going to skip over this. And so maybe there are other attributes, species attributes, that can explain some of this variation that we see. And so this is when we want to look at this ecological time as part of transcriptome variation. And so what we did is for all the species that we had transcriptome data for, we went through the literature, we mined the literature, we also talked to experts in the field, and we got a lot of social behavior, life history, and ecological variables. And Carlos helped us for some of the climate data that we get. And so we basically map all these attributes onto all these different species. And then we look at to which extent gene expression variation actually co-varies with any of these attributes within a stringent statistical framework. And so we first look at some candidate genes that we and others have identified over the years related to social behavior, as well as genes that we have previously identified in a study where we looked at the evolution of monogamy at the transcriptomic level across 450 million years of vertebrate evolution. And what we see, and this is within a phylogenetic comparative context, meaning we account statistically for phylogeny, that we actually find that things like, for example, reproductive activity and gene expression for a bunch of genes can be significantly correlated, or here precipitation and here dopamine receptor in the hippocampus. And I could show you a lot of those examples. Here's one gene that we have previously identified in the context of monogamy. Now we're looking at a completely different set of species, and we find, again, this relationship between mating system and gene expression within a phylogenetic context. Of course, genes do not operate in isolation, and so when we look at gene co-expression modules, and I'm not going to go into detail of how we do that, this is for a total of about 650 individuals across all these different species. Both for the hippocampus and the medial amygdala, we can identify gene co-expression modules that are strongly correlated with certain aspects of the social system or the ecology or the life history of these different species. And here again are some examples. Again, I don't have the time to go into details on this, but the idea is really that we can actually identify a signature of ecological variation in the transcriptome. But what does it mean, right? I've been struggling with this. What the hell is going on? Statistically, this is solid. I can guarantee you that. But what the hell does it mean, if anything? So maybe parts of the transcriptome can actually be dialed up and down in response to selection in diverse species, recruiting or redeploying similar molecular pathways repeatedly in these specific brain regions as animals adapt to different ecological contexts or evolve different kinds of mating systems. So now, and that's why we're here, let's apply this idea on the ecological timescale in the context of changing environments. And we're going to do this in the beautiful state of Texas that most of you have probably a certain idea of what its reputation is. But as it turns out, Texas is an amazing state because it has all these different ecoregions, depending how you count, they're anywhere between 12 and 17. And it has this striking, here's Austin, the red dot in the middle. We are in a short driving distance to several of these ecoregions, and they are remarkably different. It is also quite well known for this east-west precipitation gradient as well as the north-south temperature gradient. So we have a natural experiment, like a checkerboard, where we can actually now look at different species and how they have adapted to these incredibly different environments. As it turns out, Texas, especially Central Texas, is also one of the underappreciated biodiversity hotspots in North America. I mean, the species diversity is quite remarkable there, yet most of this is unprotected land. It's privately owned. So there are two fish that we are looking at now. One is the Rio Grande cichlid, which is the northernmost cichlid, the only one that is native to the United States, originally from the lower reaches of the Rio Grande River. It has large home ranges, it's monogamous, and they are really good parents, biparental. And we also look at the bluegill sunfish, which is very common across North America, especially east of the Rocky Mountains. Has male breeding colonies, so very different social system and paternal care, and much smaller home ranges. And we look at about 20 different sites where these fish are established and have become established, especially with the Rio Grande cichlid moving north, west, and east over the last 80 years or so. And now we need environmental data, okay? And so how do we assess things like temperature and water flow from data that we can actually get from satellite data, for example, from weather stations like temperature and precipitation? As it turns out, stream temperature and air temperature are highly correlated, as are water flow and effective rainfall. So we can use this as a first proxy, but long term, we'd like to get remote sensing data on even small streams and ponds. So far we get our data from different online databases like Google Earth, NASA, and so on, as long as they are available. And here for these 20 or so sites, what you see is that when we put this in a PCA, we can reduce the dimensionality of the data, and we get this north-south gradient across these sites over the course of 40 years, and we get in temperature and the east-west gradient in precipitation over the course of 40 years across these sites. And what you see is that there is huge within-site variability. So this is an added advantage of this kind of system. And we can depict this in this form. There are other ways to do this. And across species, across sites, I'm just going to show you this here. We're starting with 4 sites and how we move around. So the variability across and within sites is just stunning. We also have access to data from the Texas Commission of Environmental Water Quality, where we have data on flow rate, temperature, dissolved oxygen, pH, and so on. Not necessarily in the places where we're collecting, but nearby. Again, this is where we need better data. And finally, we track behavior using a video drone, and then use machine learning-based tracking to get at the kinematic signature of these animals, get at the ethogram, and basically infer back the classical displays that we usually quantify by observation, as well as social network metrics. Finally, we go into the brain, and we collect parts of the hypothalamus and the preoptic area, as well as the hippocampus, to compare across these sites and identify differences in gene expression in particular cell types. This is all just beginning now. That's why I don't actually have a lot of data. I'm just trying to sell you an exciting idea. And then finally, we bring a subset of these animals back from sites that are highly variable to the lab and do a common garden experiment to be able to untangle the genetics and plasticity of these different systems. So finally, we were asked to tell you what our unmet needs and desires are for the future. I think we really need to adapt machine learning-based video tracking to field conditions, and I'm not the one to do it, so somebody should do it. Effective and efficient integration of vastly different scales and modalities of open data, from remote sensing data to single-cell expression data and everything in between, into single statistical models, I think is a huge challenge. We need improved spatial and temporal resolution of remote sensing. And in terms of community-level issues, I think we need a community commitment to phylogenetic comparative approaches for causal inference. That doesn't mean we shouldn't be doing experiments. I'm all in favor of it. And somebody else said it really well earlier today. We can go to the model systems then to test these hypotheses. I refer to them as anchor species. and we need collaborations with ecosystems researchers. And with that, I want to close, and I thank you for your attention. I'd be happy to take questions. Thank you.
Wow, my head hurts. That was amazing. That was all the things I care about. Maybe I can start with a question, since you touched on a lot of really cool stuff. The use of drones to surveil behavior, can you say more about that? How do you identify species, distinguish fish species? How do you look in the water? Are they afraid of the drone flying over, etc.?
Yeah, so we've done this in Waller Creek so far, which is about 50 meters away from our lab on campus. It's a creek that runs through UT campus where these guys are native and live. And so what you see here is a pair of Rio Grande cichlids basically swimming by a lake of bluegill sunfish, and there's actually a fish in here, and probably courting another one. I don't remember exactly. And here, are two a pair, and they have much larger space range. So the drone hovers about 2 to 3 meters above the water, just high enough so it doesn't create any riffles, and the fish don't care at all. They don't seem to be worried about it at all. It moves slowly, or it doesn't move at all. It's stationary, depending on how we do it, and we're still figuring that out. The drone we use currently is kind of a relatively cheap consumer model. I would like to buy a better one in terms of the camera in particular, because that is going to be important to really get the automated tracking to work. Right now, it needs a lot of hand-holding, the machine learning-based tracking. And I mean, undergraduates are great, but I'd like to automate it and make it more reliable.
Cool.
Yes.
Any other questions for Hans? Yeah, Wolfgang.
You always have a question, Wolfie.
Sorry. This is a gigantic project, right? This is very large, and it crosses several different disciplines, from ecology to monitoring the environment to going to the gene expression measurements. How do you envision getting all of these people to talk to one another? When you said my brain hurts, I think I was right there, because it's you were moving through PCAs and giving us an abstract view of all the data. How do you envision having a structure that brings all these people together and collects all the data and puts this in a reference, so that the different people who work on this can follow?
Yeah, I appreciate that question. It's a really good question, and I've talked to some of you about this over the years. I'm not so much a fan of what I call paramilitary science or Manhattan Project type approaches for this, although they have their use, and I think the Allen Brain Institute is a great example for that. I mean, we rely on their data all the time. But this kind of approach, I think, requires kind of nimble consortia that, kind of a fission fusion type thing that change over time. And we don't have a really good framework for that. There is no funding framework for that. I've talked to people at NSF about this basically since 2008, when we started our monogamy project and so on, which took 9 years. And they just said, Hans, you're crazy, what the hell? But I do think we need to identify and build structures where people with very different backgrounds from very different disciplines can come together, work together on something, and then move on and do something else. And as opposed to having a rigid structure imposed that has to be there for 10 or 20 years. I think we need more flexibility. I call it nimble. So that's one answer to that. I think the other answer is that I'm actually not too worried about collecting all that data and so on. What I'm more worried about is that we have a lot of data already, and we don't know what to do with it, right? I was talking to Kim earlier today, and I said, I actually have a moratorium in my lab now where I don't allow anybody to do anything on more than 3 brain regions at a time, because I don't understand it. And so that's where we need people who really think about complex data much more, about how to bring AI into this and so on. So that's another aspect.
also accessing the data that's out there, right? Because often you even don't know what
Yeah.
question to ask, so that this data exists and how to get there.
Yeah, I totally agree with you. And often these platforms that house the data are not interoperable and so on, and that's a huge issue. And there are people here, and I think we'll hear about that more, who really care about this. That's huge.
Thanks.
Thank you. Great question.
Any other questions for Hans?
Well, thank you, guys. I hear there's Wiener Schnitzel for dinner. Thank you.
Okay, then let's thank Hans one more time.